Okay. The spleen. If you, this is a past stain, and out here you see the connective tissue that covers the spleen.
Here, I'll flip it so you can see.
And if you glance, just try to glance at the whole entire slide. The red part
Whatever you think.
is stained pinkish with the past.
Okay.
And the white part is this area that has these black poppy seed looking cells in it.
Here.
And in
I turn it.
I was looking for a good central arteriole. There we go. Here is one of the central arteries or arterioles, depending on how small they are. That's an arteriole. I would still call it artery. Okay? And also there is another one right here. So all this area around the arteries is called the PALS.
Okay.
The PALS contains both the T and the B zone. On this particular preparation, so far I did not see any good follicle that could be separated. There may be one primary follicle here, okay? So the closer to the, if the section is perfect, right around the artery you see the T zone, and the follicles should be somewhere out here, okay? If you can separate a B zone or follicle in the spleen. It is not as easy to separate them as in the lymph node, but if they are active, the secondary lymphoid follicles, it's easier to separate. But on this one, I don't really see any good one so far. So outside where you see this reddish staining here and more open sinuses, that is the red part of the spleen. And there is no cortex or medulla for the spleen, because these are just intertwined completely throughout the whole entire tissue. Okay, you can recognize the red part here based on the larger openings. In the white part, you don't see those larger openings because there are no open sinusoids in there. It's much more cellular. Let's, I'm going to zoom on it.
Let's see. You guys can see it.
It's more better.
Okay.
It's more fun. It's all. That's what everybody else is.
I would like to show you the sinuses in the spleen.
That looks better.
Wherever I find a
Goody.
nicer stain there.
I want a tripod. Okay.
See the veil.
Yay.
It is as good as it gets.
Every time you say drop on, I think of Minnie V. Austin Powers.
So if you look at here, this is a large opening here with plenty of different type of cells. In this area, there may be a macrophage right in the middle, and this is the sinus, okay, right here. And there is another opening here. Again, that's the sinus. Over here, this is the pulse port, right here, and the reddish stain is the basement membrane or basal lamina. And if you look at this with the oil objective, you're going to see that the basal lamina is not continuous. And that's the point of the sinus.
What do you mean? What do you mean?
Oil. I'm looking for oil.
So the Iraqis.
Oh, they have it. They have it. They are not looking for it. Those poor guys who were sent there,
They don't want to share it
they don't want to share it.
with each other.
Oh,
Oh,
Dr.
boy.
Brewer starts to talk politics here. This is a histology class.
It comes from. What are we even looking at? What slide is this?
We're in the spleen.
Oh, this is the spleen. Hot Iraq.
This is such a thick section. I never liked it very much because of its thickness.
Sure.
But if I move the
Ooh.
fine focus
A little
here
farther away.
with the oil objective, then you're gonna see
Okay.
in this area the red fibers are red staining is the basal lamina, and that is where the cells gonna migrate out into the pulp cords, and this is here a pulp cord area here. I'm gonna have to move somewhere. It's a little thinner, but maybe. And we only have one screen, right? Let me just try to find a better part of the screen. I don't like it where I was. Okay.
How many slides do we have? 3 more? Okay.
The pasting is
Hungry.
very pretty if it's not faded out. Once it's faded out, it's really hard to see what we wanna show you. All right, let's go back to the oil. Okay, if you look at here, so there is one lumen right here, and then there is another lumen right there.
Oh, this sucks.
And now, if I move the fine focus, I showed you on the electron microscope in the classroom that the cells were running like the sinuses look like a barrel, and the cells are running around these openings, and so do the basement membrane fibers. And if we cut them in cross section, you see these little dots here. That is one. It's much prettier actually in the microscope. It just doesn't really want to come in very well. So, but the